Hi everybody, and welcome to Cell-fie Science, where I'm bringing you a quick dose of science from my cell phone. I'm Jen, a PhD candidate studying plant biology, and today I want to talk to you about how do plants breathe. So you know how you breathe. You have lungs, which take in oxygen, and then you breathe out carbon dioxide, which provides your body with the gases that it needs to function. Plants also need gases to function, and they want to bring in carbon dioxide to turn it into sugars in the process of photosynthesis. And during photosynthesis, they produce oxygen, which becomes a waste product for them. So they want to release oxygen, which is very handy for us because that's what we want to breathe in. So the next time you take a deep breath and fill your lungs with oxygen, you should thank the nearest autotroph near you because that's the reason that you have this oxygen. We have lungs. What do plants have that allow them to breathe? They have a special structure called the stomata. All right, now the stomata is actually this hole, this pore, this opening right here. And it is flanked by two guard cells. And these cells can swell up, which causes the stomata to close, or they can be relaxed, which allows the stomata to be open. When the stomata are open, you have gas exchange, so carbon dioxide is brought into the leaf, oxygen is released from the leaf, and typically these are all open during the day because that's when a lot of photosynthesis is happening. The other thing that leaves the leaf during this process is water vapor. Now, water is very important to all life, and plants are no exception. When the stomata are all open, water vapor is leaving the plant and creating a gradient, which actually pulls water up from the roots to the leaf. And that helps keep pressure so that the root water comes up into the leaves. Also, though, it is losing water. So if there are really dry conditions, a drought for example, or it's super hot, then a special phytohormone called ABA causes the stomata to close. Now, you find stomata on the bottom of leaves, and I want to show you really quickly how you can observe your own stomata. So hang on for a second, and I'm going to move over to our microscope. Alright, and I'm at the microscope now. So I'm going to show you how you can make your own stomata pole and be able to observe these structures on your own. So you need a couple of things. One thing that you need is some leaves. So I went outside and picked a maple leaf off the tree by my lab. You're going to need some clear nail polish. You need gift wrapping tape, and this is really important because normal Scotch tape is frosted and you cannot see anything through it, so it has to be this really super clear gift wrapping tape. You need a slide, and you need a microscope. So what you're going to do is you're going to take a little bit of nail polish, and you're going to paint it on the underside of the leaf. And this is important because this underside of the leaf is where most of the stomata are. So what you're going to do is you're just going to take this, and you're just going to paint a little bit, and you want to avoid the veins in the midribs. See how I've painted it in between those big veins in midribs? Because that will make your stomata pole not look as nice. And then it takes a couple minutes for these things to dry, so you want to wait at least a minute while it's drying. If you don't wait long enough, then it's going to be liquidy and you'll pull up mostly nail polish and not a lot of leaf cells. So I went ahead and painted one before we started filming, and you can see there how it's nice and dry. And we're going to take a piece of gift wrapping tape, find our fingernail polish, and lay this piece of tape on there. Press it down so that it's against the fingernail polish. And then you're going to rip this tape off super fast like you're waxing eyebrows. Whip! And you can see that I transferred the fingernail polish and the leaf to the slide, to the tape. And then we're going to take the tape and put it on the slide. And now we have our very own stoma. So our stomata pole will look very similar to these two pictures that I have. This one is actually from one of the maple leaves. And you can see that there are some open and some closed. There's an open one. See how big the stoma is? And here are the closed ones. The guard cells are all swollen up and you cannot see that pore. You could turn this into an experiment with your class if you'd like by taking different times of day. Are more stomata open in the morning than at night perhaps, or every day over a series of a week, and track things like environmental temperatures or humidity. Do there seem to be more open or closed? And you can also look at stoma from different species, right? This is a coleolus plant, and this was our maple leaf. And you can see that they're not exactly the same. So you could go and do some investigating. Do they have the same number of stomata across species in the same area? And the answer is probably no. But why don't you go and find out? So that's it for this episode of Cell-fie Science, and I hope to see you next time. Bye!